Alright, hello SBI4U students and anyone else that might be watching this video. This is the first in what I'm sure will be a series of online video content in order to cover just our knowledge piece of the class. This is my first one, so it's going to be pretty awkward, but I've got some stuff set up that I think will make it a little bit easier. I'm here in isolation in my house, in my office. The fish are right there, so don't worry about the fish. They're with me. They're hanging out. So let's jump right into it. Let's talk about our learning goals for the day. Our learning goals are number one, get used to the video lectures because, like I said, this is going to be awkward. Number two, we're going to review ATP. So what is ATP? Why is it important? What kind of energy changes happen in ATP? And a general review of bond energies. We're going to review energy balance in the mitochondria, tracking those energy molecules through the steps from glycolysis down to the Krebs cycle. And then the structure and function of ATP synthase, that turbine. So this is a review of what we've done before. And because it's a review, that means that this video is not mandatory for your hours, for your content. This is just a preview video to show you what these things are going to look like and for you to be able to get used to it before we get into stuff that actually counts towards your hours. So what you'll see is I've got these learning goals that I can put up on screen. I've got the current topic that's going to stay there constantly so that you know where we are in the video and what we're talking about. And they keep in mind, I'll keep that up there all the time as well. I can edit these as we go through the video. Video lectures. Let's talk about video lectures. First off, let's talk about why video lectures. Alright, let's talk about why video lectures. Why video lectures? Well, number one, you need 10 hours of content per week. Okay, you need 10 hours of class time per week. That's a ministry guideline. So what I've decided to do there is 5 hours of video lecture. Right, 5 hours of video lecture, which is probably about a 45-minute video every day. Right, 45-minute video every day, ish. Some topics take longer, some topics take shorter. I'm going to try to keep it between 40 and 50 minutes, but I don't know. And attendance for that, will be taken through SBI quizzes. So there will be quizzes on SBI. I guess SBI is a capital E. Capital E for SBI, there we go. Attendance will be taken through SBI quizzes. Just quick, simple things, maybe multiple choice, maybe short answer, something just to tell me that you've watched the video and understood the content. The same kind of questions I ask in class. And then the other 5 hours a week, of live help sessions. Right, 5 hours a week of live help sessions. For the live help sessions, they'll be done on the Zoom platform. I know in my PDF that I sent out to everybody, I said it would be Google Hangouts, but I like Zoom more now that I've looked at its features. I especially like the fact that it tracks who's been in and out of the meeting, so it can actually get a time for everybody. So that's your live help sessions on Zoom. In order to get your 5 hours for the live help sessions, attendance there is based on logging in once a day, participating once a session. Right, you have to you have to log in at least once a day. You have to participate at least once a session. So that could be asking a question, that could be doing a practice problem with us, that could be showing off something that you drew. anything like that. So those are live help sessions. You can just get help with a project that you're working on, whatever. As long as you've interacted with me in some way, you get that that participation thing. And you do have to log in at least once a day. You have to get your 5 hours over the course of the week, which means you get to pick and choose which lecture help session you want to spend more time in. If you want to spend more than 5 hours, that's great. You know, maybe you want to hang out virtually in the virtual classroom in that meeting space and get more help. That's fine too. Yeah, but those are the requirements. That's how I've decided to run it. Why do I do this specifically? You know, for you guys, why video sessions? That's for flexibility. All right, flexibility. You guys can watch these on the weekend. You can watch them at home. You can review them. You can go over and over again. In particular, I really like the fact that I can make timestamps. I messed that up, sorry. I can make timestamps. So if you look below this video, really in that description, you can see timestamps for each individual thing. So the first timestamp will be like getting used to video lectures, like the current topic. The second timestamp will be reviewing ATP, like we have in our learning goals, and so on. And I can add other things like helpful links to other videos, stuff like that. Right, and really for the third one is I have evidence for the ministry. All right, I have evidence for the ministry that I've covered this content. So that you can actually get the credit. You know, I think we all want that. You can get the credit. Yeah. So that's the idea. Please make sure that you understand this 10 hours of content thing very well. You know, the whole 5 hours of video lecture. It's a 45-minute video every day, which, you know, if you know even a little bit of math, that's not 5 hours a week. The other 15 is to do the SBI quizzes. So I've budgeted 15 minutes of time just for you guys to hop on SBI and finish those quizzes. It probably won't take you 15 minutes, but I'm going to say it's a 15-minute time frame. And then the live help sessions on Zoom, I'm going to be keeping track of that, and I'll give you guys, I'll keep you guys updated on how many hours you have, how many hours you need, that kind of thing. Yeah. Okay. So that's the keep in mind. Well, we'll go ahead and just clear that and go back to keep in mind. All right, back to keep in mind. All right, back to the keep in mind here. This keep in mind is this video is not part of your 10 hours for next week. Keep in mind this video is not part of your 10 hours for next week. This is just a practice video. Okay, yeah, so having a look at our learning goals, we're in the get used to video lectures. We're almost done that piece. I'm just going to show off some of my camera views, and then we'll move on to reviewing ATP. We're going to review energy balance and review ATP synthase. So this lecture is really all about ATP. Let's have a look at our different views that we have. So obviously there's this view where you can see my face in full view. I can also jump over to my PowerPoint, my desktop, so you can see the ATP overview here. You know, this is just a slideshow. Right, it's just a normal slideshow. Of course, I can put my face up in the corner here, so you can see me and the slideshow at the same time. I can also stick myself over on the left side, but that's not great here. Keep myself on the right. Then there's also, I'm really excited about this, I put a camera over on this side of my screen, and that is this camera. Hello. And this camera will be used for any kind of demos if I have any kind of thing that I want to show off in, you know, bigger, wider view. You know, I might want to do some stupid, you know, hey look, this molecule looks like a triangle, and this is what it looks like. Right, I've got a wider view, and I've got this, and this is one of Miss G's whiteboards. So that's one of Miss G's whiteboards, and it's here on the table because I happen to have another camera, and that's the camera that I'm really excited about. That camera I'm excited about because it is this camera. So you see me down in the corner here, but there's a camera right up here. So there's a camera up there, and it's looking down at the whiteboard. That means I can draw. All right, I can draw things. All right, we're actually going to start with this ATP plus H2O becoming ADP plus PI. All right, we're going to have a lecture about that. So that's our ATP lecture. So yeah, these are my camera views. I'll be able to do all kinds of interesting things with them. If you have other suggestions, let me know. And I think that will take us to the end of the Getting Used to Video Lectures part of our lesson. So back to our learning goals. We should hopefully at this point be used to video lectures. Right, now it's time to review ATP. So what the heck is ATP? Why is it so important? Why do we care about it? Okay, so I'm going to change the current topic to reviewing ATP. And to keep in mind, we're going to change it to breaking bonds takes energy, making bonds releases energy. All right, so that's our keep in mind. We're talking about bond energies here, and we're going to review ATP. So ATP, if we jump over to the desktop, is the most important molecule that we track in cellular respiration. You know, we got ATP, 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 ATP. When it's created here directly from the process itself, we call that substrate level phosphorylation because it's happening directly off of the substrate. All right. So that happens here in glycolysis and here in the Krebs cycle. When it goes through NADH and FADH, these are our electron carriers, that's called oxidative phosphorylation because that's the part that's going to take oxygen. I forgot to put my camera up in the corner. Yes, that's the part that's going to take oxygen. So we'll talk about oxidative phosphorylation in detail in the first lesson back on Monday. So ATP is really important. And the reason ATP is important is specifically because of this breaking bonds takes energy, making bonds releases energy thing up here. So in order to kind of help you guys understand that, we're going to jump down to the document camera. And in the document camera here, here is the typical ATP formula that you'll see online in a lot of cases. I'm going to actually erase this and stick it up in the corner because I want to really give you guys the full thing. So often what you'll see in textbooks and stuff is you'll just see ATP down to ADP plus PI and back up to ATP, which is not great because there's actually water involved too. So please make sure that you understand that there's an H2O here, which means that when we come down, that water becomes kind of part of these molecules. When we come out, we release that water. And you're taking two things, and when you put them together, you release water. We have a name for that, and it's dehydration synthesis. And when we go from the combined thing, ATP, back to the two sub things, and we use water to break it, we have a word for that too. It's called hydrolysis. All right, so hydrolysis goes the other way. I'm going to pull up a structure of ATP here so that you guys have the structure of ATP, which I'll pull up my screen. And the slideshow. and we'll pull up a structure of ATP. Here we go, there's a good structure of ATP. Open image in new tab. There it is. So there's a structure of ATP. And what you'll see there is this part is just adenine, the A in DNA. This part here is a ribose. You can tell it's ribose instead of deoxyribose because this carbon here, carbon number 2, has an OH. So it's adenine, a ribose, and then 3 phosphates. So it's A, then triphosphate, A, phosphate, 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 ATP. So let's draw that. Let's draw that over here. And I'm going to draw it like this. You'll often see like an A. Okay, so point of order. This is the wrong way to draw it. I'm going to fix it in a second, so don't take this down. You'll often see something like this, APPP, but I find that that doesn't tell you the whole story because it's missing the oxygen bridges. When we look here, there's oxygen bridges, 2 oxygen, 3 oxygen bridges along the way. So let's actually redraw this a little bit better, where it's A to an oxygen bridge to a phosphate, to an oxygen bridge to a phosphate, to an oxygen bridge to a phosphate, right? And that phosphate has an OH on it. That, and it's going to turn into ADP, which looks like this: A to an oxygen bridge to a phosphate, to an oxygen bridge to another phosphate, to an oxygen bridge, but then that is different now, right? There's only 2 P's, there's no third P, so what's hanging off there? Well, this is where the water comes in. The water, which we can think of as HOH, OH, there's the water, HOH, is going to come in and actually break this bond, right? It's going to break this bond. And remember that breaking bonds, where am I? Here. Breaking bonds takes energy. Breaking bonds takes energy. This is going to take some energy. We'll talk about this. It doesn't actually take that much energy, and we'll talk about why. So it's going to break that bond, and it's going to install itself on the end. The H is going to install itself on this side. So where does the OH go? Well, the OH that was left over, the OH, is going to attach to where that old phosphate used to be, P. It's going to attach to that old phosphate. And we call this guy an inorganic phosphate. It's inorganic because it's not attached to a carbon thing anymore. When we look at the structure of ATP over here, you see the ribose has carbons in it, and so does the base. So all of these guys are still attached in some way to an organic molecule. But this guy no longer has any carbons attached to it. We call it an inorganic phosphate. And that's what the I over here stands for. This I stands for inorganic phosphate. All right, where does the energy come from, right? Breaking bonds takes energy. We've broken up our ATP into ADP plus P. But if you look, we've also made some bonds. So we might want to look into what we've actually broken and what we've actually made. Let's see what we break. Here we break a OP, right? A bond between an oxygen and a phosphate. Break an OP. And we also, in order to do that, if you look down here, right, it's an H and an OH, we had to break an OH bond. We had to break an OH bond. And what do we make? We made an OH bond. We make OH. And here, right there, we make an OP. So we've broken an OH and broken an OP, and we've made an OH and we've made an OP. So, according to bond energies, that should be the same amount of energy. That doesn't seem to make sense. So it's actually quite a bit more complicated than just bond energies. Oh, we forgot to put the topic on here. Huh. Oh, maybe my laptop screen is behind the topic, is ahead of the topic. Let's see. Oh, yes. Yes, it is. Okay. Sorry about that. Little issues. So yeah, current topic, reviewing ATP. Where was I? Yeah, we've made and broken the same bonds. So you're going, hey, why the heck does the energy actually change? We know for sure that this guy going this way is exergonic, meaning energy comes out, and this way is endergonic, meaning energy comes in. But bond energies don't explain it. Bond energies don't explain it. That's a really subtle thing that I'm not going to ask you questions about, but since people have this question a lot, let's explain it. One of the big reasons for it is that the phosphate looks like this. It's an A to an oxygen to a phosphate, and the phosphate has a double bond oxygen on one side and a negative charge on the other, to another oxygen to a phosphate that does the same thing, to another oxygen to a phosphate that does the same thing. Right, so one of the first things about it is all these negative charges down here, they'll repel. Right? They don't want to be close to each other. So because there's this extra repulsion, that bond, because there's the repulsion, this bond here, this bond is weaker. It's already got strain on it because it's trying to get pushed apart. So it's easier to break this bond because there's already some repulsion there. Meanwhile, if you look at the OP bond that we made, this phosphate's by itself now, so you can't get this kind of repulsion anymore. So that's one of the reasons that you have a reduction in energy. And the other piece is when we make the new phosphate bond, which I think I'm just going to get my face out of the way down here. Or actually, I'll draw it up. I'll draw it up here, I guess. Or maybe I'll move this. Yeah, I'll move this. So when we make the new phosphate bond, a single inorganic phosphate, which looks like this. A single inorganic phosphate. Basically, these negative charges can actually move around in what's called a resonance structure. These are just electrons making these bonds, and they can kind of migrate. For example, the negative charge can move up to that guy and keep that down there, and keep that OH on that side, and stuff like that. Right, there's a bunch of structures, so it's always kind of moving those negative charges around, keeping it spread out, so that it doesn't it doesn't stay on the same atom for too long, that negative charge. There's probably some kind of analogy here, like when you have to hold something hot, like a hot potato, and instead of just holding it in your hand, you're kind of like doing one of these. That's what's going on with the negative charges here, is that because they're constantly moving around, essentially you get a lower energy molecule here than you do here. Here there's only two places for it to jump. Here there's like 4 places for it to jump. So it's a lower energy bond, meaning that we release more kinetic energy when we do it. So that's the two things. Is number one, there's this negative charge repelling it, and number two, this resonance structure stabilizes the molecule. So even though we've made and broken the same bonds, this oxygen phosphorus bond is a lower energy than this oxygen-phosphorus bond, because this one is trying to hold together against this repulsion, and this one doesn't, and this one has those resonance structures. So that's your ATP. That's where the actual energy comes from. It's a pretty good amount of energy. It's not quite, making a new bond kind of energy, but it's somewhere in something like 70 kilojoules per mole, which is pretty good for just reshuffling some bonds. Looking at the energy diagram for this, if we wanted to make an energy diagram, oh, oops, that's too low. An energy diagram for this. Energy diagram. We might have ATP here. We have to break bonds to come up, and then we make bonds to come down. ATP plus PI. And so it moves back and forth. And this delta H, the delta H, is something like minus 50 kilojoules per mole. Something like that. Minus 50. It's kind of fuzzy down there. Anyway, it's minus 50 kilojoules per mole. So that's ATP. That's what ATP. That's the energetics of ATP. But to kind of help explain why this doesn't happen all the time, why it doesn't just break apart all the time, and how it's used for cellular processes, I've got a video that I'm going to show you about ATP and muscle cells. So let me cue that up, and we'll explain how ATP can actually produce all of this stuff. So back to this camera view. I'm going to pull that up. It's called the cross-bridge cycle. And we'll find that. I had that. Yeah, here we go. Sorry about that. I should have just paused the video while I did that. I forgot I can pause the video. So one thing let's while we're waiting for that to load, let's keep in mind
Les fromages râpés Petit Québec, mélangés pour le Québec. Les Petits Québec, tellement nous.
Making bonds releases kinetic energy. Right, the energy that's released is actually kinetic. It jiggles stuff around. It makes things move, and that's how this thing can move. So our topic then here is ATP and muscle contraction. So our current topic is ATP and muscle contraction. Let's jump over to this view. This is the video I'm going to show you guys. We've got the captions on. In order to show you this video, I'm going to mute my microphone, or else we'll get feedback, and that's not going to be fun. So I'm going to mute my microphone. We're going to watch the video, and then I'll go through it with you. So.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
Okay, we're back off of mute. So yeah, that's kind of showing you how ATP binds to muscle cells and makes the muscles move. And it's actually just kind of like a series of steps, and each step, as the muscle contracts, requires one of those ATP power strokes. They don't go backwards. They can't forcibly relax. They actually just kind of come apart. But one of the most important things here, and this is probably going to be part of our keep in mind, ATP won't release energy unless ADP has somewhere to go. Alright, so ATP won't release energy unless ADP has somewhere to go. And you saw that here in this picture. Normally, when the troponin is active, it is blocking the binding sites. But when the calcium is in, that's when your nerve cells tell your muscles what to do. It opens up these binding sites, and now the ADP and the myosin has somewhere to be. And that's why your muscles aren't just constantly contracting, even though they're full of ATP. They need somewhere for the ADP to go. So it's a constant cycle of making and breaking bonds. You always want to have a new bond able to be made before you can break a bond. So that's kind of just a general idea here. Where the ATP comes in is here. Right? The ATP comes in here, and what it does is, as it comes in and binds to the muscle cell, it turns into ADP plus P, and that releases kinetic energy. It's kinetic energy that gets released. And kinetic energy, the energy of motion, what it does is it actually just pulls the head of the muscle, the myosin head, back. So it creates a tension in the head. And then as it comes off of there, and you have the actin here, the tension gets released. But the ATP is the thing that's actually pulling it back, and then it gets released. And so over the course of thousands and thousands of these processes, your muscles contract. Everything I'm doing here, talking, moving my fingers, all of that is all done with thousands and thousands of these cycles happening very, very, very quickly. So that's ATP. We've covered ATP. So going to our learning goals, we have reviewed ATP. We've talked about how the ATP is where the energy comes from, which is these that the inorganic phosphate is a lower energy molecule than the phosphate when it's attached to ADP for a variety of reasons. We talked about the role of water here, and we've talked about the general energy stuff. About 50 kilojoules per mole gets released. So that's ATP. That's why we care about it so much. That's why it's so important. Now, our next thing is our energy balance inside and outside the mitochondria. Where do the energy molecules come from? Where does the ATP get formed? And all that stuff. So we're going to jump back over to this view. We're going to pull away the learning goals, and I'm going to change the current topic. The current topic is now going to be energy molecules in the cell and mitochondria. All right, those important energy molecules. And what we want to keep in mind is where does each process take place? That's our keep in mind. Where does each process take place? Yeah, so what we've learned so far is that glucose turns into pyruvate through a process called glycolysis, and there's actually two arrows here. Well, there's a few arrows here. We've talked about the energy investment phase and the energy output phase. We'll cover that. Now the pyruvate turns into acetyl-CoA. This happens inside the mitochondria. And then the acetyl-CoA is the input to the Krebs cycle. And the Krebs cycle, as you can see, is where most of the possible ATP gets generated. And we've also talked about how only 4 ATP gets generated. Only 4 ATP, 4 ATP, this one and this one, 4 ATP gets generated in the directly from the substrate, so substrate level phosphorylation, and the other 32 ATP, those get generated in the mitochondria using oxidative phosphorylation, which we haven't really talked about yet. So let's jump over to our document camera, pull up the screen, and we're going to go through the process all together. Yeah, that was good. Okay, so let's draw the processes in black. So our first we'll first call it glycolysis 1 for the investment phase. We'll have glycolysis 2. Glycolysis 2 for the payoff phase. Then from there it goes through pyruvate, oxidation, and then the Krebs cycle. All right, pyruvate oxidation and the Krebs cycle. And what are the molecules? Well, we're obviously going to start with glucose. All right, glucose goes through glycolysis 1. It's a series of 5 steps. What comes out is 2 G3P molecules, G3P. Now the P in G3P is a phosphate. It's a phosphate attached to it, just like the P from ATP. So that's a good hint for you that this requires, so I put a minus there, 2 ATP. Now what I mean there is that the ATP turns into ADP plus P. That P actually ends up here on the G3P. So that's the energy investment phase. Then there's a payoff phase, so that G3P, that 2 G3P turns into 2 pyruvate after it comes through glycolysis 2. And it's a payoff phase because we make 4 ATP. We make 4 ATP. We also, at the same time, make 2 NADH, which will go off into the oxidative phosphorylation later when we're talking about ATP synthase. And we make 2 water. We make 2 waters as well as part of this process. Yeah, that's the payoff phase. So pyruvate comes out, two pyruvates come out. The pyruvate is going to go through pyruvate oxidation, and it turns into acetyl-CoA, two of them, two acetyl-CoAs. And at this point, it's useful to think about how many carbons we're dealing with during this cycle. So glucose is obviously a 6-carbon molecule. G3P are each a 3-carbon molecule, so the carbon number hasn't changed. And pyruvate is also a 3-carbon molecule, so the carbon number hasn't changed. But acetyl-CoA, your cognate there is acetic acid or vinegar. It's got 2 carbons, so it's a 2-carbon structure, which means that we've lost 1 carbon for each pyruvate, or 2 CO2s overall. So there's a -2 CO2 here, or I guess +2 because we're making 2 CO2s. And when we make a CO2, each CO2 we make, we actually also make an NADH. So there's 2 NADHs that get made during pyruvate oxidation as well. That's pyruvate oxidation. Now the acetyl-CoA goes into the Krebs cycle, and the Krebs cycle is a cycle. The Krebs cycle is a cycle. It's got 8 steps classically, and the acetyl-CoA just feeds in as the input to the Krebs cycle, and it's got 2 carbons. If it was constantly feeding in with its 2 carbons, the cycle would just gain carbon over and over and over and over and over again. So those 2 carbons get pulled out, and it's 2 carbons per acetyl-CoA, which means that total we make 4 CO2s as we pull out those carbons. Also per glucose here, we make 6 NADHs at various points in the cycle. We have 3 different points we make NADH. We also make at 1 point, which means twice per glucose, we make an FADH, FADH2. And we make, at 1 point in the cycle, a GTP, which is very much the same thing as ATP. G is just guanine, and A is adenine, and they're very similar structures. They're both purines, so they have that double ring structure. So yeah, GTP is basically ATP. So we're making 2 ATPs. And we need, that's a minus, we need at 1 point in the cycle 2 waters, which nicely contrasts the 2 waters we made up here. We use 2H2O minus 2H2O, which cancels out the 2H2O we use up here. So that's your basic overview of what we've got so far. This is what you guys are responsible for on the test when we have it. The other thing you're responsible for is, as shown up here, where does each process take place? Where does each process take place? Well, let's talk about that. This is all one cell. You can imagine the whiteboard is one cell, which means that there's cytoplasm around. So all of this happens in the cytoplasm. In the cytoplasm of the cell. The cytoplasm. But there is also the mitochondria. So let's show off where the mitochondria is. The mitochondria is here. And the mitochondria actually has 2 membranes. So here we'll say this is mitochondria. That's the mitochondria. It has 2 membranes. So the 2 membranes are the inner and outer membrane. The inner membrane is folded, as you know. The inner membrane is folded. So that means that pyruvate oxidation and the Krebs cycle both happen inside the matrix of the mitochondria. So everything in here is considered the matrix, right? The matrix of the mitochondria. This part here is the intermembrane space. none of our glucose processing happens there. None of the carbon stuff happens there. All of the CO2 gets released inside the matrix, and that's just, all the CO2 gets released inside the matrix. So that's the overview of all of our energy molecules so far. That's what things look like and where they happen. You're responsible for literally this entire whiteboard diagram here. If you want to just draw it yourself, that might be a good way to study and to remember, to make sure that you know what happens and where and which energy molecules are involved. All right, let's talk about what happens to this NADH and FADH stuff, because that is something that's important for us. So let's jump back over here. Go back to our learning goals. We have reviewed energy balance inside and outside the mitochondria, which means now the last thing is just talking about where those NADH and FADHs are going to go. We're just going to do a general brief overview of ATP synthase, and where it is and why it's important that it's there. So yeah, let's do that. Changing our current topic to ATP synthase. Current topic is ATP synthase, and the keep in mind is why does ATP synthase sit on the membrane? Right? Why does ATP synthase sit on the membrane is our keep in mind. So let's have a look. I'm just going to pull up the picture of it. Oop, that's not it. Here's the picture. Right, here's the picture of ATP synthase. It goes through a process called chemiosmosis. You might know osmosis as water moving from low concentration to high concentration of water, or from low concentration of solute to high concentration of solute, rather. Sorry. Chemiosmosis is chemicals moving from high to low. So you see that there's a high concentration of H plus on one side and a low concentration of H plus on the other side. The analogy that I used in class, was of a hydro dam. So let's do that. Let's by analogy talk about a hydro dam. So I will pull up a new whiteboard and talk about how a hydro dam works. In a hydro dam, what you have is a reservoir with a wall in it, and on one side of the reservoir you have high water, right? High water on one side and low water on the other. And what they've got is there's a way for the water to go through. Now, if that way was just completely open, you know what? I'm going to shrink down there we go. Low water on the other side. Now, if that was just open, all the high water would flood through, and a bunch of energy would come out, and you wouldn't be able to get any electricity out because you don't have anything in the way. So we put something in the way. We put a turbine in the way. A turbine. That way, as the water tries to flow through, we slow it down with the turbine, and as the turbine slows down, we get energy out because the turbine is impeding the flow. The same thing happens in the mitochondria. Let's diagram the mitochondria. We have two membranes. These are like the walls over here because things can't go through them. And the high water is actually a high concentration of H+. And that H+ is all throughout this intermembrane space. So it's in the intermembrane space. There's lots and lots of it. Intermembrane space. Lots and lots of H plus in the intermembrane space, and not a lot of H plus, you know, maybe there's a few in the matrix. So this is the mitochondria itself. And outside here is the cytoplasm. So this is why the mitochondria needs two membranes, because it wants to hold a high concentration here, which means it needs a membrane stopping these from leaking out to the cytoplasm, and a membrane to stop it from leaking in to the matrix. Now in here is the Krebs cycle. So all of the NADH, a lot of the NADH and FADH2 are generated in here through the Krebs cycle. And part of the work of the mitochondria is actually replenishing the H plus gradient by grabbing those hydrogens off of those NADHs and FADHs and pushing them up. The other thing that NADH and FADH carry with them are electrons. So electrons come up with it. These electrons, these high energy electrons that come off, help push the hydrogen ions across because they're negative and these are positive. So they almost like help them carry along by neutralizing the charge. Those electrons have to come back down where they want to be up here, but you know, they want to be up here with the positives, but we force them back down, and we also pull in these hydrogens back out. And where they go is they actually get attracted to oxygen, and that turns into water, which leaves. That's why it's called oxidative phosphorylation, because there's a redox reaction that happens at the end where the oxygen accepts the final electrons. We're constantly regenerating H plus off of NADH and FADH, going onto oxygen, and out it goes. So we're building up this crazy gradient up here. That gradient has a name. It's called an electrochemical gradient. So there's this electrochemical gradient that's trying to push the H pluses in. Just like we have a gravity gradient trying to push our water in. And there's a turbine in the way, and that turbine is what you see in your note on this side here. That is ATP synthase. And to put it briefly, every time one hydrogen flows through ATP synthase, you get one ADP plus P turning into ADP. And it's basically done almost like a pair of pliers where as the hydrogen flows through, you have ATP on one side and P on the other, and just slams together, literally just slamming together each time. But it's done in an actual turbine, an actual rotating sense. I'll put up a video under this video, so look down below. You'll see the link to that video. You'll have a full view of what actually happens. But that's the important piece. So you guys are also responsible for knowing where the gradient is, where the high concentration is, what electrochemical gradient actually means, where ATP synthase sits, and why it's important for it to sit on the membrane. Okay. Also, how do we recharge the hydrogen gradient? So we recharge it by using the electrons from the NADH and FADH, also the hydrogens from the NADH and FADH, to constantly add hydrogens up here, and we're also constantly pulling away hydrogens by making H2O and kicking it out. Yeah, so I think that covers everything. We have hopefully gotten used to video lectures. We have reviewed what ATP actually is. We reviewed energy balance, the basic scheme of what happens in cellular respiration, and then the structure and function of ATP synthase. Monday's video will go into we'll go into this here, like this specific part here, how those hydrogens are pulled up off of the NADH and FADH2. There's a sequence of pumps that does that. But for now, that's the end of this video. It's a bit longer than I thought because I wanted these to be 45 minutes long, but I guess we spent 10 minutes just talking about video lectures, so that's about the level of content that you can expect from me. Thank you very much, and I'm signing off on my first video. Thanks.